Hello, good afternoon.
Hi. Hi, Florian. Can you hear me?
Yes, and you?
Yes, yes. Sorry about that. I have no
No
idea.
worry.
It was just not letting me in.
I think we need to pause the recording, right?
Sorry?
We need to pause the recording while we try to get everything up and running. Isn't it?
Yes, yes. Okay, uh, welcome everyone, and sorry for the slight delay, which was caused by some technical issues. Um, Uh, Without further ado, I think I'll introduce our speaker today. So this is a presentation on behalf of the Royal Society of Chemistry, Process Chemistry and Technology Group. And we're very pleased today to introduce Florian Medina, who works for Johnson and Johnson. Um, and due to the technical issues, I'm afraid I didn't get a chance to check with Florian's biography. So maybe, Florian, if you're happy to introduce yourself,
Sure.
that would be great. Florian will speak. If you have any questions, please feel free to put them in the chat. And then, Florian, hopefully you're happy to address the questions at the end.
Sure, I will. Thanks, Charlie, for
Okay,
the
so
kind
thank you.
introduction.
Thanks, Florian. Over to you.
Yeah, do you hear me well? All good?
I can hear you clearly.
Perfect. Right. Good afternoon, all. Maybe a quick introduction then from my side. Thanks, Charlie, and the entire RSC team for giving me the opportunity to be here today to present this work. I guess as you can hear, I'm French, and I might have quite a strong accent for some of you, so please bear with me. And if you have any question, don't hesitate to shoot at the end of the presentation or write it in the chat during the presentation. As you can see on the slide, I'm a HT chemist now at G&G. I started there about 10 years ago as a process chemist. I worked there for about 5 years as a process chemist and then moved to the HT team. The project I'm going to talk about today is actually about a project that where I was working as a process chemist. That was the last project I worked on as a process chemist. And you will see that even on the simple API, you can have a lot of, a lot of fun from the synthesis point of view. So I will be giving few slides, for few corporate slides about G&G so that you have some introduction on the company. And then we'll go to the deep part of the chemistry. I will be able to share after the presentation, the slide on the chemistry part, not the corporate slides. All right. So quickly, corporate slides. If I'm able to get the slide to move, voila. Welcome at G&G. For those who know the company, we're a pharma company. We've been funded in 18, wait, I'm gonna move this one. Oh,
Not not seeing your
yeah.
slides yet, Florian.
So wait, I'm trying because I do have this one. This is bothering me. Hopla. Is it any better like this? Is it moving?
No, we're still. I'm still just seeing you, I think. Wait a minute. No, Florian Medina's screen. Yes.
Okay, maybe if you don't mind, I will switch off then my camera so that I get most of the Wi-Fi connection. Voila, that should be hopefully better. So about G&G quickly. So the company was founded in 1886 by the brothers Janssen. And I guess in Europe, most of the people knows Janssen Pharmaceutica. And to give you a bit of a background, this company, Janssen Pharmaceutica, was acquired by G&G back in the early 60s. And since then, we are part of this American company. We went through a branding exercise a couple of years ago, and now Janssen Pharmaceutica doesn't exist anymore. We are all part of G&G, and the entire pharma part is under G&G Innovative Medicines. Um, this is not working again, sorry. The company is strongly driven by our credo, which is our moral compass. Um, and this moral compass is here to make sure that everything we do is to make sure that we do things safely, with integrity and ethically, so that we're all on the same pace within the company. Few numbers. I think it's also important to realize how big G&G is. It's a very large company. If I can get more, yeah. As you can see, a lot of employees working for this company. The headquarters is based in the US, in New Jersey. And another number I like to give to introduce the company is investment in R&D. As you can see, it's a massive investment from the company, and it's important to hear this kind of number, I think. As said, so the Janssen Pharmaceutica doesn't exist. Now we are mainly working in two different parts at G&G. So you have all the innovative medicine, which is everything about pharma. And the second part is about medtech, which is all the medical device and so on. And obviously, I'm working for the innovative medicine part, and a bit more specifically in the small molecule business. So welcome at the development department of G&G. As you can see, here is a picture of our main R&D site, which is based in Belgium, in between, let's say, in the middle of Antwerp and Eindhoven, a bit at the border in between Belgium and the Netherlands. Most of the development department is located on these sites, but we also have entities in the US, in China, of course, and also in Switzerland. This department is mainly involved during the clinical trials period. We start to work on the project usually in the phase 1, and we stopped after the registration when the compound reaches market. And to give you a bit more detail, usually we start to really invest in the project as soon as the compound starts the phase 2. That's where we start to have internal FTEs that start to work on the project. All right. Chemistry. So here is a compound I would like to talk about today. As you can see, it's a rather, if I may, simple API, a small molecule, only one chiral fragment, which is also a bit easier to synthesize, I guess. I won't discuss much about the application or the indication of this compound. I'm a regular chemist, and all the biology part is a bit more difficult for me, so I prefer to focus on the chemistry part. As explained on the previous slide, we started to work on this project mainly during the phase 2A. That's where we started to have internal FT working on this project. And to give you an order of magnitude, we were 2 chemists working on the project at the beginning, and we ended up at 3 chemists. When we started to work on this project, this is that was the GMP route at the moment, and I will walk you through it. So the first step is a methylation using dimethyl carbonate as a methylating agent, quite a nice reaction. Then you have a direct trans amidation with LHEMDS as base. You get to this amide intermediate, and after you have a sulfonylation, activation, and finally a coupling to get to the API. So it's quite a nice synthesis, linear synthesis, but it's difficult to make a convergent synthesis from such a small molecule. But I will walk you through some of the issues we noticed during the different GMP campaigns. So first of all, during the methylation step, using dimethyl carbonate, one of the side products of this reaction is methanol. And because of the methanol produced during the reaction, we saw that the reaction was stalling and not able to reach full conversion. So that was the first issue that needed to be tackled. The second one, in the transamidation step, the base was used in a large excess, and LHNS is pretty expensive, and that was not really nice from this perspective as well. And finally, here in the third step, the sulfonylating agents, we had some issues with the quality, and because of this low quality, we are having a lot of impurities forming in the process, and we're in need of developing a thorough crystallization, and that's why we had such a low yield. In terms of processability, so here this compound, this intermediate, this amide, was very insoluble in toluene, and the crystallization was pretty difficult to control. So we had a lot of issues, and actually the analysis was also insoluble in toluene. So that was a slurry to slurry, not very nice in terms of process development. So that's why we wanted to modify this part to have a better control on the crystallization. Here, this sulfonic acid compound was also problematic because it was hygroscopic. And for the next step, the activation, that was a problem because any traces of water was a problem, as you can imagine. And finally, all the problem was the use of in two steps of DMF here in the first step and in the activation with also with DCM. Those solvent of concern was also problem, two issues that were needed to be solved. So in terms of strategy now, to give you a bit more detail on what we wanted to do, if you consider all the issues I've listed just here, most of them are in the early part of the synthesis. Surprisingly, they've started the synthesis without using the N-methylpyrrole. So for us, it was a bit of a no-brainer. We should start directly for the N-methylpyrrole so that you avoid this step, this methylation step. One of the good things from this synthesis, it was the late introduction of the chiral building blocks. This one was introduced in the last step, so that was very good from the process point of view. So that's something we wanted to keep. And as a classic, we also had some time constraint. Usually you have only a few months to redevelop a synthesis, and here, in that case, with only two chemists, we could not spend too much time redeveloping the entire synthesis. So we decided, because of all these points, to focus mainly on the early part of the synthesis, and actually we wanted mainly to change the synthesis toward this key intermediate, this amide intermediate. What we've done at the beginning was mainly focusing on different type of acylation of N-methylpyrrole, but it was not very productive, till the point where we look at how our starting material from the previous GMP step were made. So you start from this NH pyrrole, you do a trichloroacetylation, it works well at reflux in deethylator, and then you quench it with sodium hydroxide in esterification using the haloform-type reaction. So this works well, and we directly thought, okay, if we use the N-methylpyrrole, it should work. We look at the literature, and indeed, it was possible. So we come up with this first proposal. So you start from N-methylpyrrole, you do trichloroacetylation, you quench it with hydroxide. So this is the regular haloform reaction. You form the carboxylic acid, and then you make an amide coupling. So that reaction should work. It's almost textbook reaction, so that should be fairly simple. So the first step, trichloroacetylation, and indeed this one works pretty well. It works well, especially in ether solvents. So in MTB, it's a direct reaction. It's done within 2 hours at room temperature. What we've seen upon scale up is that from the safety point of view, we have a bit of accumulation of the acetyl chloride compound. That's a classic, and to solve that as a classic as well, we were doing slow addition of the trichloroacetyl chloride, and we increased a bit the temperature to 45 degrees so that we were able to control the accumulation during the reaction. In terms of crystallization, actually this compound was very simple to crystallize. We found out that a mixture from alcohol and water was giving very large rods, very simple to filter. So we are very pleased with the filtration, a bit like brown sugar. It was working very well. The cake was very good. And we're able to quickly isolate a gram of this compound to study the downstream chemistry. And I said, yeah, earlier, any type of acylation with other acylating agent on the N-methylpyrrole was not working. We also tried to use PTC condition and so on, but nothing really worked back in the time. And actually, we started to have already the first issue. So we did few scale-ups, as said, to support the downstream chemistry. And what we noticed is that the amount of compound isolated was fluctuating. The IC was always very high, as you can see here on the slide, but the amount obtained was fluctuating. And we started to look a bit more into that detail because it was a bit surprising that you had a variation of plus or minus 10%. And actually what we figured out was this trichloroketone was able to sublime upon drying, something we clearly haven't anticipated back in the days, but this one was subliming in the vacuum oven, which meant that no isolation was possible. Of course, you can do a workup, and that's something we implemented later on, but it was not possible in the view of a production campaign. What we did was still to produce some solid from this crystallization using, you remember, the mixture of alcohol and it was methanol with water as anti-solvents to develop the root coating effort. What you need to remember is that this trichloroketone is an activated ketone. So as soon as you have a base and traces of water or alcohol, like what you can get from the crystallization of this compound, will lead to the formation of the corresponding acid or methyl ester if you have traces of methanol coming from the haloform-type reaction. Okay. So now we are able to get to that compound, to the trichloro intermediate. Let's look at the next step, which is the amide coupling. So we started to play with classical coupling agent condition, and here we are very surprised. We were mainly getting messy reaction, low product formation. It was very difficult to obtain the desired product with high conversion. For example, we tried on the CDI coupling the trick from this OPRD where you had acetic acid trying to activate the acylimidazolium formed by the activation with CDI and the carboxylic acid, and that never worked. So we thought that finally, first, this aniline was clearly a bad nucleophile, so that may explain why the reaction is not very working. But the info from the low reactivity using CEI may come from the low reactivity of this carboxylic acid. So we thought, okay, let's try directly to activate this compound, forming the acyl chloride intermediate. And here again, if I may say, Nuff is low, kick back again. This reaction didn't work. As soon as we were adding oxalic chloride or thionyl chloride, we were observing black precipitation, a very dark reaction mixture, and decomposition of the mixture very quickly. So we tried to understand what was the root cause of this decomposition, and we thought that it may be due to the acidic condition, and that one of the two compounds here might not be stable under this very acidic condition. So we thought if we use the carboxylate salt instead of the carboxylic acid, we may be able to buffer a bit the reaction condition and still be able to perform the reaction. And here it worked. We are very pleased to see that with thionyl chloride, we were able to form the desired acyl chloride. And here, just to show you, if you use oxalic chloride, actually, we are not able to obtain the reaction. We did not observe any decomposition, but we are forming this compound upon quench with diethylamine. Anyway, we're able to get our activated carboxylic acid. Let's look at the next step, which is amide bond formation. Oh yeah, sorry, forgot that one. Of course, as we were making carboxylate salt, those compounds are known to be hygroscopic, so we looked at that quickly in the lab. So we did a 10-gram synthesis of each of them and look at the one which was the less hygroscopic in that case, and it was the case of the lithium salt here, the top row. Back in the days, the lithium was not so much in the hype because of the battery industry, so we were okay to use this type of carboxylate salt, but I think today that would be a different story. But still, we made this lithium salt and moved forward. We did first a scale up to prove that the reaction was working, so we started from about 20 grams of the desired N-methylpyrrole. We did trichloroacetylation. We introduced a water wash of to remove the HCl side product of the acetylation, and then the solvent switch to acetonitrile. Here, the solvent switch is pretty interesting because this second step reaction works well in acetonitrile, but also with this distillation, we are doing an azeotropic drying of our reaction mixture. We obtain the desired salt with high yield and high purity with a decent PMI of 52 over 2 steps. Amide bond formation. So here the reaction works very well in acetonitrile. We could make it work with different type of base. Surprisingly, with organic base like triethylamine, we are obtaining a very thick slurry, so we didn't move forward with that, and the best candidate was in the end potassium carbonate. And the best option was actually to do a reverse addition. So here we are adding the acetyl chloride compound to a solution containing our aniline and the base. And like this, we're able to obtain 80% isolated yield with very high purity again. So here we have our first proof of concept. We have a first route tool that was leading to our desired intermediate. We are happy, but if you look at the synthesis, and if you're honest, it's a bit redundant. So first you synthesize this activated carboxylic acid, this trichloroketone. You quench it to make the carboxylic acid. You reactivate and quench again to make the desired amide. So here we're thinking maybe we should be a bit more lean in this synthesis. Maybe we should try to be a bit more efficient, and especially can we directly quench this trichloroketone with this aniline? We looked at the literature, and actually the only example we found in the literature was with a very good nucleophile, especially alkylamines. This reaction works and it is described. But with aniline or even electron-poor aniline, there is nothing described in the literature. So we thought that might be a bit more challenging than what we thought. So we started to study this reaction still, and we first look at the reaction with organic base. Of course, if you make this reaction with inorganic base like potassium carbonate, phosphate, or even alcoholate, you will generate the corresponding carboxylic acid, or in case of alcoholate with the corresponding ester, right? So that's why we mainly focus on organic base. And here we are very pleased to see that with DBU, at high temperature, we are able to obtain decent, almost full consumption of the trichloroketone and formation of the desired product in about 70%. So we are very pleased, and we started to see if we could work under using smoothest reaction condition. And actually, at 50 degrees, the reaction works okay-ish, if I may say, in different solvents. The best one is still acetonitrile, and toluene also is a good one. But if you recall previously, toluene is not a good candidate because this compound is fairly unstable, insoluble, sorry, in the reaction mixture, as well as this aniline. And indeed, you will see later in the table, I'm a bit ahead of my slides, sorry. So in dry toluene, we only have low isolated yield because the compound crashes directly, and it's very difficult to isolate pure material and correct material. We also look here in the middle of the slide using catalytic amount of base, but here it doesn't work. As you can see, the reaction is still heavy. Even with longer reaction time, we are stuck at 70% conversion using substoichiometric amount of base. The best candidate was finally DBU with an excess, 2 equivalents in dry acetonitrile, and we were able to obtain the desired compound with 79% isolated yield. So we started to have a proof of concept. So we did a scale up here. So what we did was, again, this trichloroacetylation. We have a solvent switch from acetonitrile, MTB, sorry, to acetonitrile. We added first the aniline so that everything was dried and we could directly start the haloform-type amidation, the idea of having the entire mixture dry so that you don't form any carboxylic acid that could quench the DBU. We spent a lot of time working on the isolation. It was not very straightforward. We tried a lot of workup, crystallization, and finally, none of them were successful. And the best finally come by doing HCl quench of the DBU, and then you charge water as anti-solvent and you crystallize your desired compound. As you can see here on the outcome, it works pretty well. We have a good isolated yield and a very high purity. The nice part is also the PMI is very low over these 2 steps, and we're able to obtain the desired intermediate in only 2 steps. But still, could we make this approach a bit more attractive? Because actually, if you consider using 2 equivalent of DBU here, and that you need to quench afterwards, it's not very elegant. So we thought maybe there is a better way to do this reaction. Maybe we could make it a bit more efficient. So we looked back at what we've done at the beginning of this project, and we mainly focus on DBU, and we haven't tried any other strong base. And actually, if you try other, I listed here in the table other type of strong base with their nucleophilicity based from the Meyer group work, as well as their basicity. And if you look a bit in the detail, DBU and DBN even they give about the same type of reactivity. If you look, the strength of the base is about the same, as well as the nucleophilicity. If you move to a very strong base like MTBD or TBD, there it's very difficult. TBD gives much higher reactivity with higher, more basic base and a more nucleophilic base. But in the meantime, methyl TBD is much less active than DBU, while it's more basic. So here, clearly, you need to have a kind of a buffer in between, a mixture between having a strong base, but also quite a strong nucleophilicity of your base to make the reaction to happen. We also look at other very strong base like P2H, phosphazine base, works well as well, but a bit lower. So that's good for us because P2H are very expensive as base, so we didn't want it to go there. And actually, potassium dibutoxide worked if you do a very specific addition order for your reaction. As soon as you move from this addition order, it didn't work, and we're mainly getting the terbutyl ester. The nice part is that as we are having very high reactivities entry 8 with TBD, we thought maybe we could start to use stoichiometric amounts. And here we are pleased to see that indeed, using substoichiometric amount of TBD, entry 12 and 13, we are able to get the desired product with satisfactory yield. And actually, we have the same activity if we use 2 equivalent of DBU or 0.2 equivalent of TBD. So what we think here is that TBD is actually playing the role as an acid transfer catalyst. We discussed with our computational colleagues, and here is what they come up with. So TBD is able to activate both the aniline partner and the trichloroketone. So you make this kind of intermediate, and that's why you're able to get a good reactivity and especially a catalytic activity. From the calculation, it matched what we observed in the lab. So DBU is a good candidate, better than M-methyl TBD, but TBD is way better in terms of activity. And this type of dual activation is actually already reported in the literature. You have here a JACS paper where you have the same type of dual activity by TBD. All right, now it's time for the proof of concept using catalytic amount of base for this haloform-type amidation. Of course, here we modified a bit the first step because any trace of acid or water in the first step would kill the base here because you would form from the haloform reaction the carboxylic acid and that trap your base. So that's why we needed to implement a bicarbonate which is in the first step. After that, we have the solvent switch from MTB to acetonitrile, strict control of water as well, so that's very good. And we're able to perform the reaction, and as you can see, we obtain isolated yield of 80% with a high purity. So we are pretty pleased because we are able to reach the same activity with DBU, but to be honest, we don't have any improvement compared to the DBU process. We are able to have same type of purity and the same level of isolated yield. So no clear improvement, if I may say. So for that, we looked at how can we improve the crystallization of this second step, the haloform-type amidation. So first, we look at the solubility of our product in different solvents. And here, if you look at acetonitrile, normally the solubility is pretty low. So two candidates with lower solubility are toluene and MTBE. And if you remember, in toluene, the product and the aniline are not soluble, and you will form a slurry to slurry, which is usually quite difficult to control. So we didn't want it to go in that direction. MTBD could be a good candidate, but still we wanted to understand first why acetonitrile, even though the product has a solubility, we are not being able to push more product out of the Mosel liquor. So we did some very specific solubility tests. We tested different additives to our reaction mixture to understand what was solubilizing our compound. So chloroform is a side product from the reaction, so this might have an impact on the solubility, and you see there is a small increase in solubility. If you charge water, actually water is not a very good anti-solvent at that moment. And the important point here that if you can, please keep it on the back of your mind, is that if you had a second volume of water here, you have a phase separation. So because of the chloroform, we think it's because of the chloroform, you're not able to add more water in this reaction mixture and then you have a layer separation. But here, more tests were done, and that's where we were able to understand what happened in this mixture. So as soon as you have TBD, TBD increases a lot the solubility of our compound, you see a tremendous increase. And even though we quench TBD with different acids, so acetic acid or HCl, we keep very high solubility of the desired intermediates in the reaction mixture. So that's explained why we are not able to push more product out of this mixture, and we always have about 10 to 15% product lost in the Mosel liquors. So for us, we thought, okay, if we go back to the first table, maybe the best candidate is MTBE because that's the only one that has a lower solubility for our product. And that's what we tried first to investigate. So for that, if you remember, the first step is done in MTBE, so that's fairly convenient, but still there is some work to do. HCl is a side product of this trichloroacetylation step. So it means that we need to remove this one without using water because water and MTBE do not form an azeotrope, and we wouldn't be able to dry the MTBE solution after an aqueous workup. So here the first solution was maybe doing a distillation to remove the HCl. We are also using a bicarbonate wash, you remember, to quench and to remove any traces of quenched acetyl chloride. And here the idea was, could we invert the stoichiometry to solve this issue? So here I meant, can we use an excess of methylpyrrole in comparison to the trichloroacetyl chloride, so that all the trichloroacetyl chloride is consumed to make the trichloroketone. So that's what we tried, and actually it worked. We are able to obtain the desired trichloroketone with a very low amount of residual HCl in the mixture. So we pushed this stock solution to the desired intermediate, so the haloform-type amidation, and here the reaction works. It needs a bit of adjustments, like a slow addition of the trichloroketone, because in MTB, the solubility of this intermediate is very low. You have a very fast crystallization. So we had to increase a bit the temperature to avoid some crusting, but still we are able to obtain the desired product. Unfortunately, if you look here, the purity was not that great. We lost about 10 points in purity, and that was quite of a problem. Of course, if you do a rework, you dissolve your solid in acetonitrile and then you add charged water, you're able to obtain what you want, but it's not very elegant and you add an extra step. So we wanted still to improve this direct crystallization. So we went back to our solubility table, and actually, if you remember, the chloroform here brings some issues. As soon as you have chloroform, acetonitrile, and water, at some point, if you keep adding water, you're not able to add more water, you have a phase separation, and then you're a bit stuck at this quantity of water, quantity of antisolvent you add to your reaction mixture. So we saw that maybe if we were able to remove the chloroform, we could add more water to our reaction mixture. And that came actually at the right moment where chloroform was an issue in the production environment. At that moment, we are starting to work towards a pilot plant campaign using this process. And actually, the engineers came back to us, watch out, guys, your molecule contains chloroform and is strongly acidic. That's something we cannot do in the plants. So if you remember, at the end of the haloform-type amidation, we quench the DBU with HCl. We don't do a pH control quench, and what's is happening is that we end up with a strongly acidic composition. If we want to dispose this mother liquor, it has to be done. It can be done, but it needs to be neutralized to pH 4. So for that, what we can do, you add a base to neutralize the excess HCl, and you reach the right pH value. Unfortunately, this waste layer is not in a reactor, it's in a tank, so you cannot charge carbonate, for example. If you charge carbonate, you will generate CO2, and if you want to control the CO2 release, you need to have a stir tank, which is not a waste tank. So that's why they start to add sodium hydroxide, but the problem is that with sodium hydroxide, you may form carbene in the presence by the reaction of sodium hydroxide and chloroform. We tried a lot to isolate any of these carbene species. We never been able to, but that's not enough for production people. They were really concerned about this safety risk. So they decided, they pushed us towards the fact that we need to remove the chloroform. And that was actually a good thing if you consider that might have been an issue for the isolation of our compound. So here what we've done, we went to our engineer colleague and we discussed with them trying to find a solution to remove these chloroforms from the reaction mixture. We ended up with 3 potential solutions. The first one was membrane filtration. That could have been very challenging because if you look at the size of chloroform is fairly close from our compound and other intermediates in the reaction mixture. So this one was probably difficult. And also, most of the time, the membrane, you only have one supplier, and that's something we tend to dislike in the company, a single supplier for any type of equipment or intermediate in the synthesis. Acid-base extraction was not possible. The compound was not soluble in acetonitrile, so we would try to do then an acid-base extraction with slurry, so that's clearly not possible. And finally, the last option that was remaining on the list was the distillation. So here we ask our colleagues, our engineers colleague, can you try to use models to see if we would be able to remove the chloroform from this acetonitrile mixture? And actually, doing calculations, they were able to find out that you do have an azeotropic mixture in between acetonitrile and chloroform. And then we ask them, what if you have a bit of water? Well, actually, with water, as you can see around on this scheme, on this graph. If you add water to the reaction mixture, you have an even better azeotrope mixture, and you're able to remove more of the chloroform. So that's very convenient for us. And that's something we started to look a bit more into the detail. All right, give me just a second, I will drink a bit of water. All right. Sorry. So here we should be able to do an azeotropic distillation from acetonitrile and chloroform and remove like this most of the chloroform from our reaction mixture. Different option possible. Shall we do this distillation before or after the HCl quench of the base? So here I'm talking about the workup in the haloform-type amidation. So on the left part of the graph here of this scheme, you have the standard workup. So you quench your base with HCl, no distillation, and you do the crystallization by adding water and filtration, and you obtain your product. You have a lot of chloroform, more than 5 weight percent in the mosel liquor, and you have traces in the washes as well, and in the cake. If we do now the chloroform removal by distillation after the quench, so first you charge HCl, you quench your base, you do the distillation, and then crystallization by adding water. As you can see, the isolated yield is about the same. The amount of chloroform in the isolated product is much lower, so that's a good point. And also in the mosel liquor or in the washes is also much lower. If we compare now with first doing the distillation of the chloroform and then doing the quench of the base with HCl, actually we end up about with the same values in terms of isolated product, 3. And the value of chloroform in the mosel liquor or in the washes. So it was a bit difficult to decide which was the best option. And actually, the solution came from looking here at all the impurities. With this last option, what we figured out is that most of the impurities formed during the process were decomposed during this procedure. And we thought here, if we need to then find all these new impurities and in the process of understanding the formation of the impurities and what would be the quantities we could purge in this process, that would be a very difficult exercise. So we thought that this option of first doing the quench of the base and then distillation of the chloroform would be the best, so that we would be able to control most of our impurities. So from that, we went back to our engineers and we discussed with them what would be the best scenario actually to remove the chloroform. Shall we do constant volume distillation? So you keep the volume in your reactor constant and you keep feeding with acetonitrile while you're doing the distillation, or you do a classical put and take or take and put, and so that you remove by fraction your solvents. So I will walk you through this complex graph. So the most important part is actually the red line. So the dotted line here is the constant volume scenario, and the plain line is the put and take approach. And as you can see, the plain line is going is below the other one. So with this option, we're able to remove more chloroform. So here, the put and take scenario should be better than the constant volume distillation. And if you look at the consumption of solvent, so here that's the green line, sorry, the dotted line is the constant volume distillation. So we have a constant feed of acetonitrile in the tank. And if you look at the put and take, we do see that at some point we have a significant difference in terms of solvent consumption. So we decided to move forward with the put and take scenario. We needed to do a bit of adjustments. We also set the target of 1000 ppm of chloroform in the mosel liquor. So that was coming from guidelines like ICH guideline and so on. And the best scenario was actually to have 2 addition of acetonitrile and finally an addition of water as antisolvent to compensate with the water that was coming from the HCl and removed during the distillation. And we also did a second modification in the first step, so the trichloroacetylation. So here we did a constant volume distillation this time. Here again, it was a discussion with our engineers, and this one seems to be more efficient, so we went for that approach. We made a confirmation at 0.5 mole scale. So again, we compare prediction and actual value measured in the reactor. So you see the plain red line is the predicted value amount of chloroform in the tank, and you see the cross is the one that we measured. So we are pleased to see that the model we are fitting actually is the measured value of chloroform. And in terms of isolation here, as you can see, we're able to boost the isolated yield. So this time we're at about 87%. The amount in the molecular is much more under control, so we gain about 10 points. And also the amount of chloroform in the molecular is much more controlled so that it could be disposed properly. Unfortunately, of course, if you modify the composition of your reaction mixture, the crystallization change. So that's what we've seen, and actually the previous proof of concept was very difficult to filter. It takes quite some time, and as you can see here on the picture, we mainly obtain flakes that tend to aggregate, and then the filtration is very bad. As a standard, this was the original process. You see, we have large rod, very easy to filter. And of course, when it's like this, we start to discuss with engineers, they want to do a dairy. Unfortunately, this time, the dairy approach was not really a success to improve the crystallization. And what we found out was actually, as you can see here on the picture, we were able to slightly improve the shape of the crystals by having a longer NGK time and a slower cooling crystallization, which seems to be obvious, but yeah, probably here the dairy was not properly set it up and we were not able to define properly the range for this crystallization. All right. I'm reaching the end of this presentation, and now the idea is to compare the different processes. So here, that was the first, so that's the final process with chloroform removal. And here, the first thing we did was to apply this process to the DBU. DBU was in the pilot plant, and the idea was also trying to improve this work. And as you can see here, now we're able to obtain the desired product on the kg scale. With a high isolated yield of about 80%, very high purity, so no losses in the purity. And the nice part is that we're able to have a control of the losses of the product in the molecular, and the amount of chloroform is much more under control also in the molecular. We have a slight increase of the PMI. Now we are at 35, but that's because of the removal of the chloroform, because you need to add more acetonitrile to perform this removal. So if you compare with the original DBU process, you have a clear improvement in the isolated yield, about 10 points, which is coming from the molecular mainly, at the cost of the PMI, as just said. And now if we compare with the TBD process, you remember the catalytic version of this haloform-type amidation. So here, with this removal of the chloroform, we're able to have also a very high yield, this time slightly above 90%, very high purity as well, comparable to the previous one, and a lower PMI, mainly due to the fact that we use a catalytic amount of base, which is then quenched by a catalytic amount of HCl. But still here, we had a lot of discussion, and in the end, we prefer to move forward with the DBO process, mainly because TBD was difficult to source. And also in terms of quality, sometimes we had a few batches with variable quality of the TBD, and that was very problematic for the reaction. So we decided to finally move with the DBU process for the pilot plant campaign and the production campaign. All right, now it's time to conclude. So we are able finally in this project to develop a new and innovative ligand synthesis for Bersacapavir, and it was mainly based on this haloform-type amidation. We also were able to develop a catalytic version, of this reaction based on the unique reactivity of TBD. Here, if you remember, we mainly think it's because of this acid transfer ability. Sorry, let me rephrase this acid transfer catalyst ability, and that's why we think this reaction can happen using catalytic amount of the base. In the last part of the talk, I've shown that predictive tools are very useful if you want to work on a workup and how to transfer a simple reaction to the production environment. And of course, something I haven't mentioned, it's a great teamwork. It's not only the two chemists involved in the project, there was many more people. And we are very happy to publish this work earlier this year. Now it's time for acknowledgements. Thanks to the team that worked on that. It was a very nice project and very great team here. Thank you for your attention, and as well, thank you to the RSC team. I'm happy to take any as well.
Thank you very much, Florian, for a really fascinating piece of process chemistry. I think a very elegant articulation of how what can appear to be, quite well, at first sight, relatively straightforward chemistry can have hidden depths and hidden challenges, especially when you're developing a process. As I mentioned, if you have any questions, please do type them in the meeting chat, and I'll feed them to Florian. In the meantime, I just had a couple of questions. The first one, I think you talked something about timelines, and I just wondered roughly how long did this new process development take from beginning to end? Because I think you were talking about having only a limited window to work in. So can you give an indication of roughly how long this development work took before you were able to effectively pass the final process on?
Yeah, thanks, Harry. Very good question. So actually, what usually we try to do in that case, as soon as possible, we try to implement the new chemistry on different scale. So once we were able to get a proof of concept on the haloform-type amidation, we went quickly to the mini plant, pilot plant, and so on and so forth. So in terms of time, we usually have between 3 to 6 months to implement any new reactivity to have a first proof of concept on gram scale, kilo scale, and then mini plant. So usually about 6 months, we need to be able to run at least a kilo lab experiment on that new chemistry.
Great, thanks. Oh, there's a question just come in. The question is, at what point did it become clear that the supply of TBD would be difficult, and hence driving the use of DBU as the base?
It's a very good question. Thanks. So this one came very late, actually. So because to be frank, the production team were very pleased with the DBU process. It was running in the pilot plant on 80 kg scale with like a charm, no issues, and they were very happy with it. And on our side, we liked the TBD process. It was a bit leaner, better PMI. So we tried to push it more and more, but still at some point we had to take a decision. And that's why we went so far into the scale up of this TBD process to be able to compare peer and peer, right? So we pushed actually this project very far. Some people might I think that we could have stopped it a bit earlier, but we are really believing that this one was much better. And in the end, if you look really at the purities, the isolated, and so on and so forth, the TBD has a marginal impact. Also in terms of cost, in the end, 2 equivalent of DBU has about the price of 0.2 equivalent of TBD. So even here in terms of cost, the gain was not clear.
Yeah, and I can sympathize. In my times working in directly in process chemistry, I had similar issues where we were from the lab end, we were pushing what appeared to be better chemistry, but the manufacturing facility were often, their constraints are somewhat different, and yeah, better chemistry doesn't always mean it's going to get
Exactly.
you one to take. Couple of more questions have just come in. So the first one, what methodology was used to measure the solubility of the amide?
Wait, maybe I can leave if I can. Can I move the slide back? Hopla, maybe I can move directly to that slide. So I guess it's that slide.
Yes, the isolation, I'm assuming.
So there is two approach actually on this slide. So on the left part of the slide, the first table is classical solubility measurement, which is, if I remember well, it's solubility curve building, and where you're able to measure what is the limit of solubility of your compound. Those type of measurements are very slow because you need to do a kinetic solubilization of your compound. So this first table is actually done at our colleague from analytical development. And then on the right side, this one was done by us in the lab where we're trying to see what type of additive was driving the weird behavior in the crystallization. And simply you start in a tube, you have a known volume of your of solvent, sorry, and you charge little by little your compound until you're not able to dissolve it. And that's why we're able to reach those numbers. They are less accurate, of course, than the classical solubility measurement, but at least it gives you a good idea on the solubility and a good idea on the range for the solubility of your compound in these mixtures.
Yeah, yeah. So they gave you the right order of magnitude to make
Exactly.
decisions.
Yeah, yeah. And you see here it's clear, right? Chloroform has marginal impact. First volume of water is the same. As soon as you get TBD, then it's completely different. So that's why we wanted to understand what was the factor that was driving this high solubility of our compound in the reaction mixture.
Great, thanks, Florian. Next question: is the process sensitive to mass transfer or energy transfer issues? So, or heat transfer, I assume they mean by that. Were you able to use just a regular reactor setup, or did you have to do anything special in terms of mixing or heat removal for this process?
It's a good question, thanks. Mass transfer, it was, I think, clearly a problem in the first process when they were using toluene, and that's why we wanted to step away. So as soon as you have a strong crystallization with very low solubility of your compound, the mass transfer is very problematic because you have direct crystallization, and if you don't have a good steering, then you have a lot of issues. You can start to capture part of your material. In our case, as we moved to acetonitrile with higher solubility, it was much better. In terms of mass transfer, I don't remember that we had some issues.
You you mean heat transfer?
The heat transfer, sorry.
Yeah.
No, heat transfer was okay. And I think here is more a safety-related question then. Sorry, I misunderstood.
Yeah.
My bad. No, it was okay. Actually, it's quite surprising because the TBD addition had a little impact on the second step. The first step was a bit more problematic, as the classic acetylation are a bit more, I would say, the heat release by acylation reaction are a bit higher. But that's a classical reaction. So we know that you need to do a slow addition, you increase the temperature, and then you're fine.
Yeah. And I'm guessing you have your process safety people looking at that sort of thing.
Oh, yeah.
Straight off. Yeah. Yeah. Um, that's the last of the questions. I actually have one more myself.
Yes.
You, you obviously had a lot of input from your engineers in terms of modeling of the distillations in particular.
Yep.
Were there any other examples within this where modeling played any significant role, or what was distillation really the key input of modeling in this case?
We did, I think, 3 other DOE. So for the DOE, mainly we rely on our engineers. Internally, we use Jump software, and it's a bit of a difficult one to play if you don't have the good background. We have a good understanding on the DOE, but having the engineer to be able to play and extract the entire data from the DOE is a bit easier for us. So here they helped a lot. And in terms of model on that one, back in the day, we didn't have so many models. I think nowadays we would do more on the pKa determination. So we have much more on that now. Well, this project is about 5 years old, so that's also why back in the days we didn't have so much available.
Sure, sure. So the distillation modeling was really the key,
Yeah.
the key modeling enabler in this one, though. Yeah.
Mm-hmm. Yeah. And actually, to come back on one of the previous question, we mainly try to use the same reactor. It's a multipurpose reactor in production, and whatever we do has to fit in that reactor. So sometimes we can play a bit with the model to better understand what is the impact of the steering if you have a thick slurry, but most of the time it works pretty well. So we are not able to modify those reactors. So that's maybe something I missed. I haven't replied earlier.
So again, the process needs to be adjusted to the plant
Yeah,
as much as possible. Yeah, that makes again, it's very familiar to me. So
classic in
yeah,
pharma, yes.
yeah, exactly. General purpose plants are pretty much the standard. Okay, I can't see any more questions coming in, so I think at this stage, just like to thank Florian once again for an excellent, very elegant example of process chemistry, showing how you can both clean up reactions, but also, I guess, how sometimes pragmatism and external forces are controlling. You maybe can't always do what you really want. You're being driven by what external stakeholders require of you as much as what you do, and chemists therefore need to be flexible to be able to deliver what their end users are asking for them. So I think with that, yeah, thank you very much, Florian,
Thanks to you
for
guys.
your talk. And if anyone does have any further questions,